Hello everyone, welcome you all in the part 2 of gene therapy. In the part 1 we understood the various components of the gene therapy including vectors. In the part 2 we will discuss about the technologies where all these components are going to be utilized, and we'll see how the gene therapy can be done. So we see the technologies as well as we'll see the applications of gene therapy. So here first we'll discuss about the approaches for correcting the faulty genes. So which are the common approaches we have for correcting the faulty genes? So the first one is the very common approach where a normal gene may be inserted into a non-specific location within the genome to replace a non-functional gene. So this is usually a common objective we have, common approach we have, that whatever the non-functional gene is there, we have to replace it with the normal gene. Second is an abnormal gene could be swapped for a normal gene through homologous recombination. So this is another approach. We just a kind of swapping, interchange kind of situation we can create for a normal gene through homologous recombination. Next is the abnormal gene could be repaired through selective reverse mutation, which returns the gene into its normal function. This is like a repair. We are not going to substitute an entire gene or we bring out the new gene into it, while we reverse the mutation process and then bring the gene into the normal condition. Next approach is the regulation of a particular gene could be altered. So the degree to which a gene is turned on or off means if any one gene which is been behaving abnormally and creating a lot of serious issues in the body, so maybe that gene can be turned off or switched off for some time, right? So that these problems can be sought out, or those adverse difficulties which are coming in can be kept on hold for a time being. So these are some approaches we are following. Now we'll see there are two types of gene therapy are there. First is called as the somatic gene therapy, another is called as the germline gene therapy. So what is this somatic gene therapy? Involves is introducing a good gene into targeted cells with the end results of treating the patient, but not the patient's future children, because these genes do not get passed along to offspring. What do you mean by it? Once a person has been treated by the gene, or it has been altered, gene has been replaced by that, but these genes which have been replaced or altered in this particular human being will not be inherited to the offsprings. So it's for a one lifetime you are treating for the genetic disorder of that. This is the form of gene therapy that is being done at most genetic laboratories throughout the world. Next is the germline gene therapy. What is happening here? Injecting foreign genes into fertilized eggs or in sperms producing cells, which will then pass any genetic changes to future generations as well. So this is really sensitive matter, where changes which you are making in to a particular person will be inherited, will go, these changes will continue to the generations. This form of gene therapy is extremely controversial, and currently very little research is being done in this area, both for technical and ethical reasons. Now you can understand there are a lot many ethical reasons behind this if you want to carry it out. So these are the two different type of gene therapies are there. Now we'll see which are the methods need to apply for the gene therapy. So there are various methods. One is called physical method, direct injection of DNA method, liposome mediated DNA transfer, calcium phosphate transfection, electroporation, retrovirus vectors, other viral vectors, targeted gene transfer via receptors, artificial chromosomes, site-directed recombination, activation of genes of related function. About the transfection, we have already discussed in the video of gene cloning. Now we'll see here the first method or the first technology which is being used for this is called physical method to enhance delivery. Under this we have a method called electroporation. Electroporation is a method that uses short pulses of high voltage to carry DNA across the cell membrane. This shock is thought to cause temporary formation of pores in the cell membrane, allowing DNA molecule to pass through. So here they are using high voltage pulses to take the DNA, and the DNA should cross the membrane and reach inside to the cell where you are actually wanting this DNA to be placed. And because of this high voltage what they are carrying it creates a pore, and by that pore in the membrane, and then the DNA will enter into those pores. However, a high rate of cell death following electroporation has limited its use, including clinical application, because one way or other you are just making damage to the cell wall, cell membrane, and that damage may be severe. If that damage is severe, the cell death will be the result. So this is a one limitation of electroporation. Second method is gene gun. I think gene gun method you have already seen in the gene cloning method. The use of particle bombardment or the gene gun is another physical method of DNA transfection. In this technique, DNA is coated with the gold particles and loaded into a device which generates a force to achieve penetration of DNA into the cells. So here, by using a one device called as gene gun, under that you load they load the DNA, and then they fire it, and then it goes directly in with a high speed into the cells. If the DNA is integrated in the wrong place in the genome, for example in a tumor suppressor gene, it could induce a tumor. So another complication with the gene gun is that it is so fast and rapid that it may place your DNA into the wrong places, and if it goes into the wrong places, maybe the results will be different. So it may cause the tumor or something else. Another method is called sonoporation. Sonoporation uses ultrasonic frequencies to deliver DNA into cells. So here they are using ultrasonic frequencies. The process of acoustic cavitation is thought to disrupt the cell membrane and allow DNA to move into cells. So by the different method of transport or different force applied by the ultrasonic methods, it takes the DNA into it. Next is the magnetofaction. In a method termed magnetofaction, DNA is complex to a magnetic particle. So here what is happening, they are treating DNA with a magnetic particle, and a magnet is placed underneath the tissue, culture dish, to bring DNA complexes into contact with the cell monolayer. Right. So by using the magnet, they are trying to take the DNA into the cell. Now these were the physical methods. Now we'll see the chemical method is there. First one is called oligonucleotide. The use of synthetic oligonucleotide in gene therapy is to inactivate the genes involved in the disease process. So here they are using the oligonucleotide technique to inactivate that particular, is kind of switch off process, what we have discussed, switch on switch off process. So here by some chemical they are trying to inactivate those particular gene which are abnormal and creating lot many bad conditions. Next is the lipoplexes and polyplexes. To improve the delivery of the new DNA into the cell, the DNA must be protected from damage. So these are the synthetic chemical which are used to protect the DNA, because this is something you have genetically engineered it, and you have the DNA which you are going to insert into that particular gene or even DNA where you want to replace that particular gene. So it should be properly protected. If it is gone wrong or it get damaged before it inserted into that, it may harm in a other bad way. So for that purpose, these lipoplexes and polyplexes have been utilized. So initially anionic and neutral lipids were used for the construction of lipoplexes for synthetic vectors. Next is the dendrimers. A dendrimer is a highly branched macromolecule with a spherical shape. In a particular, it is possible to construct a cationic dendrimer, that is one with a positive surface charge. When in the presence of genetic material such as DNA or RNA, charge complementarily leads to a temporary association of the nucleic acid with the cationic dendrimer. Right? So first you create dendrimer, and then this dendrimer, which is having a positive charge, right? What you have created, then it will associate with the nucleic acid. Means the nucleic acid and the dendrimer will get associated, and these charges will help it out to allow it to enter into the cell. And because here the charges are utilized, so the membranes potential or membranes what called the static forces, electrostatic forces have been utilized on this purpose, and by using endocytosis it takes in. Next method is the hybrid method. Virosomes are one example of these hybrid methods. They combine liposomes with an inactivated HIV or influenza virus. This has been shown to have more efficient gene transfer in respiratory epithelial cells than either viral or liposomal methods alone. Other methods involving mixing other viral vectors with cationic lipids or hybridizing viruses. So here hybrid methods are utilized, where uh, your delivery system being loaded with the, or you can say complexed with the viruses, and viruses are having a convenient method to enter into the cells, so they are utilizing their own method and get into it by using the liposomes combined with it. Another method is electrical methods. Electro transfer are more well established. Applying an electrical field to cells alters the resting membrane potential, which can induce permeability through the formation of reversible structure membrane changes. Like that may cause the electropores. So these are the electrical methods can also be utilized. So here you have seen that these are the technologies or the methods used for the gene therapy. So any one of the method which is suitable for that particular gene and that particular situation, that particular cell, they utilize it and perform the gene therapy method. Now, once the gene therapy method has been understood and been practiced, where we can utilize them. So we'll see the applications of gene therapy. Gene therapy is likely to have the greatest success with the disease that are caused by single gene defects. This is an important point to know. We always prefer to treat, or you can say still we are limited in that area. So because of that limited capability, we are able to treat maximally those disease which are those who are caused by the single genes. By the end of 1993, gene therapy had been approved for use on such diseases as severe combined immune deficiency, familial hypercholesterolemia, cystic fibrosis, and Gaucher's disease. Treatment of cancer and AIDS also been there. Numerous disorders are discussed as candidate for gene therapy. Parkinson's, Alzheimer's, arthritis, heart diseases. So these are all discussed that it can also be treated by the gene therapy one day. Criteria for selection of disease for human gene therapy. So we have these 5 criteria. The disease is an incurable life-threatening disease. So if the disease is life-threatening, means ultimately patient is going to die. So there is a chance that you can give a last chance by using the gene therapy. Because of the gene therapy itself is having lot many threats. So we have to choose it with the proper justification. Second, organ, tissue and cell types affected by the disease have been identified. So if you know exactly what disease and what organ, what tissue is being affected, so clear identification is needed. The normal counterpart of the defective gene has been isolated and cloned. And the thing is that the normal counterpart of the defective gene has been isolated and cloned. Means already you have identified, then after identification you have cloned it, isolated it and cloned it. Then only you can do the further things. If suppose that in some cases isolation is not possible or cloning is not possible, then you can't do this. The normal gene can be introduced into a substantial subfraction of the cells from the affected tissue. Or normal gene, if normal gene can be introduced, then the means subfraction of the cells, if that feasibility is available, right, that affected tissue is giving you this freedom, then it's possible. The gene can be expressed adequately, and from beginning we are discussing the adequacy or the what called the expression of the gene. That's why they are using retroviruses and all. So gene the gene expression should be adequate, then only you will get the results. Techniques are available to verify the safety of the procedure, and also if you are using the method, and you should be having all the information related that, and with all the safety available for that method. There is one disease, mucocutaneous gene therapy. So in these mucocutaneous related diseases, gene therapy can be applied. Transient expression of naked plasmid DNA could be used as the local treatment of various skin and mucosal lesions where the corresponding gene product has therapeutic or immunization potential. So there are diseases which are skin related diseases and mucus lesions. If you are replacing a gene out of it and giving a new gene into it, and that new gene will produce these certain kind of proteins, and these proteins will heal up those processes, these lesions, and solve that problem. If that's happening, by this method they can treat the mucocutaneous diseases. Herpes viral infection. So here there are herpes viruses. So herpes viral infections, there are lot many herpes viral infections are there, for which also we can do this treatment. Epstein-Barr virus diseases, Epstein-Barr virus diseases are also there. Hemoglobinopathies or thalassemia, this is one of the targeted disease. Replacement with the normal functional beta globin gene, and in some cases with gamma globin gene. So that these diseases are happening because of the disrupt beta globin and gamma globin genes, so that we are replacing it here. Another is hemophilia A and hemophilia B. Another is very well known and very well discussed disease is the cystic fibrosis. Is a common severe autosomal recessive genetic disease which is caused by disinfection of epithelial cell surface, CAMP activated chloride channel. So this is a disease which has been caused by the problem in the communication pathway. So if there is a dysfunction of the CMP activated chloride channel in the epithelial cell, then the cystic fibrosis has been caused. Another is the HIV infection. It's also been used for the HIV infections. Another disease, this is also belongs to the skin diseases, that is gene therapy for the oral squamous cell carcinoma. So in this OSCC disease also the gene therapy can be applied. Gene therapy in periodontics. So in lot of oral and dental diseases also the gene therapy can be utilized. So gene therapy is quite well used in the dentistry. Gene therapy for cystic fibrosis lung diseases. Already I have discussed about cystic fibrosis. Next is the Parkinson disease. In the Parkinson disease, you can see that progress achieved in the realm of gene therapy over the past decade has offered solution to many of the delivery constraints, and several aspects of PD presented overly as an ideal clinical indication to target using GT. What are these clinical implications? The well-defined, localizable, and targetable neuronal system involved with major motor symptoms. Second is the need for relatively small titer and volume of vector targeted to those sites, which avoid the systemic circulation of immunogenic materials. And third, the large and increasing demand of improved therapeutics with an aging population, with the whole bolsters impact and financial support for research and development. So Parkinson's, these are the progressive degenerative disorders, neuronal degenerative disorders, which have no treatment. So in these cases where the treatment is not available, treatment is available, but we can say the cure is not available. So if the cure is not there, the gene therapy is one of the method to apply for this. Next is the gene therapy for infectious diseases. Already we have seen some viral diseases. Gene therapy for arthritis is also there, where arthritis is also, this is one of the idiopathic disease. We don't know the real cause behind the arthritis. So if the arthritis and a big number of population in the world affected with the arthritis, especially the old people. So if the gene therapy is offering the solution for it, is one of the wonderful curing method. Genes can be delivered locally at the site of disease pathology, such as the joint, by intra-articular injection. This is the way they do it. They just inject it into that area. Alternatively, therapeutic genes can be delivered using specific circulating cell types such as T cells, or antigen presenting cells such as dendritic cells. So this way in arthritis it can be treated. Gene therapy also used in the diabetic neuropathy. In the diabetic neuropathy, patients are getting severely affected by the neuronal degeneration. So this can also be treated by the gene therapy method. So usually the target cells for the gene therapy uh, are given below, like peripheral blood lymphocytes. So the diseases related to these cells, are means can be applied the gene therapy on them. So peripheral blood lymphocytes, hematopoietic stem cells, fibroblasts, hepatocytes, keratinocytes, skeletal muscle myoblasts, airway, epithelial cells, vascular endothelial cells, tumor cells. Now I'll talk about the advantages and disadvantages of gene therapy. So here are some advantages. Germline gene therapy offers a true cure, and not simply palliative or symptomatic treatment. So here it's not a symptomatic treatment, it is not just a palliative treatment, but it's a cure. Germline gene therapy may be the only effective way of addressing some genetic diseases. By preventing the transmission of diseases genes, the expense and risk of somatic cell therapy for multiple generation is avoided. So this one way we can see, like once the disease can be cured, the generations and generations can be secured by these diseases. Medicine should respond to the reproductive health needs of prospective parent at risk for transmitting serious genetic diseases. So if there are the parents who are going to transmit these diseases to their kids, at least can be controlled. The scientific community has a right to free inquiry within the bound of acceptable human research. You'll see the disadvantages are also there. Germline gene therapy experiments would involve too much scientific uncertainty and clinical risk, and the long-term effects of such therapy are unknown. Such gene therapy would open the door to attempts at altering human traits not associated with disease, which could exacerbate problems of social discrimination. There are reports that few of the countries have, without any ethical consideration or without any notice or permissions, they have done these kind of genetical changes and tried on human beings. There are some reports on China and initially on Japan, who have they have tried on these genetic changes. Right now, there is a body which controls all this, but even though there are the places where the secretly these kind of researches are going on. A germline gene therapy involves research on early embryos and affects their offspring. Such research essentially creates generations of unconsenting research subjects. So this is a big problem. Gene therapy is very expensive and will never be cost effective enough to merit high social priority. Germline gene therapy would violate the rights of subsequent generations to inherit a genetic endowment that has not been intentionally modified. So this will always be the therapy of the rich people, and a lot of high cost is involved. So it never going to be the treatment modality for the common people. So these are the disadvantages of gene therapy. So by this, I would like to uh, end my session, just to add few points on ethical issues which are there surrounding on the gene therapy. Because gene therapy involves making changes to the body's set of basic instructions, it raises many unique ethical concerns. How can good and bad uses of gene therapy be distinguished? Who will decide the good and the bad, right? So that's a good and bad body has been made by its own set of instructional communication methods. So by changing a gene, you are changing the entire set of instruction. So here, if anything is good or what is bad, nobody can decide. Who decides which traits are normal and which constitutes a disability or disorder? Will the high cost of gene therapy make it available only to the wealthy? So then also one social divide will create. Could the widespread use of gene therapy make society less accepting of people who are different? Yes, of course, because once these experiments are happening, then people will get change of their disabilities, and maybe the changes can be of the another level, and they can be of a different kind of people. Now you know that if Avengers are living next to your home, in movies it looks good, but if they are living next to your home, it's very difficult to accept them. If the Hulk is your neighbor, so it's not that easy to going to happen. So its acceptance will be very, very difficult. Should people be allowed to use gene therapy to enhance basic human traits such as height, intelligence, or athletic ability? Then nobody will be different from each other. Everyone can remove their what you believe is a uh, disability, or you believe is a shortcoming in you. You can get modified. Nobody will be smaller in height. Nobody will be less intelligent than anybody else. Everybody is having all the good aesthetic abilities. So these all are the difficult situations which can happen. So these are the ethical issues actually. And if you really want to read more, there is one more topic in this further. That's called is the gene therapy safe? So the aim must be to maintain the virus in its latent period as long as possible, and to protect uninfected cells from viral infection, and perhaps to enhance the immune response against viruses. So that's what, like in some cases it can be good, in some cases it cannot be good, like that is the case. These are some more gene therapy is there in applications. It can be applied in the cancer treatment as well. Also it is applied for other viral vaccinations. And if you want to read more, there is a one short information about the future of the gene therapy. So I hope you all have understood what the gene therapy is, what are the components of gene therapy, what are the methods applied for the gene therapy, and what are the applications, advantages, disadvantages, and the ethical issues behind the gene therapy process. So these are the references I have used as I have shown you before. So please go through with all this and try to get this whole process of the gene therapy. Thank you all for learning honestly. Thank you.